The major enzyme in glycogenolysis is glycogen phosphorylase. Recall that in glycogen structure, most of the glucose molecules are connected in linear fashion by 1,4-glycosidic bonds. And the enzyme that cleaves 1,4-glycosidic bond is glycogen phosphorylase. That's why glycogen phosphorylase is considered the major enzyme in glycogenolysis. Because if glycogen phosphorylase is not working, glycogen cannot be touched. Because of its importance, glycogen phosphorylase has to be regulated. The concept is that in both liver and muscle tissues, glycogen phosphorylase is regulated by phosphorylation-dephosphorylation mechanism. Glycogen phosphorylase is activated only in phosphorylated state. So phosphorylation activates glycogen phosphorylase. Phosphorylation and thereby activation is provided by glycogen phosphorylase kinase. And dephosphorylation, thereby inactivation, is provided by phosphoprotein phosphatase. In activated state, glycogen phosphorylase cleaves 1,4-glycosidic bond. This results in release of glucose 1-phosphate molecule from glycogen. Then cell converts glucose 1-phosphate into glucose 6-phosphate. And now there is huge difference between muscle and liver tissue. Muscle tissue uses glucose molecules only for own needs, to provide energy for muscle contraction. But liver tissue is the major organ that maintains blood glucose level. Because liver tissue has glucose 6-phosphatase that cleaves phosphate group that results in formation of a free glucose molecule. And free glucose molecule can exit into the circulation, thereby increasing blood glucose level. And because these two tissues have so different functions, they have different regulation of glycogen phosphorylase kinase. In the liver tissue, phosphorylase kinase is activated by glucagon and epinephrine. So why these hormones stimulate phosphorylase kinase? Let's assume the person in a fasting state. In fasting state, glucose level in the blood decreases, and the first hormone that reacts on low blood glucose level is glucagon. Pancreatic alpha cells increase glucagon secretion, so glucagon level in the blood increase. Epinephrine is a hormone that increases during intense physical exercises. And with physical exercises, muscle tissue uptake a lot of glucose molecules from the blood, because muscle tissue uses glucose in glycolysis that will provide energy for muscle contractions. Obviously, the higher the uptake of glucose by muscle tissue, the lower becomes glucose level in the blood. So to prevent hypoglycemia during physical exercises, the nature created this epinephrine-mediated stimulation of glycogenolysis. And also decreasing blood glucose level during physical exercises stimulates secretion of glucagon. So in this case we have a synergy of these two hormones. So in fasting state or during intense physical exercises, the consumption of glucose prevails over glucose income. So the goal here is to provide additional glucose income into the blood. And this vital glucose income will be provided by glycogenolysis in the liver tissue. Because recall that liver can release free glucose molecules into the blood and muscle tissue cannot. So what is the mechanism of action? Glucagon acts on glucagon receptor in the liver tissue and epinephrine acts on beta 1, 2, 3 receptors in the liver tissue. Activation of these receptors through GS protein causes activation of adenylate cyclase that in turn converts ATP into cyclic AMP. And increase in cyclic AMP activates protein kinase A. In turn, protein kinase A activates glycogen phosphorylase kinase, and activation of glycogen phosphorylase kinase is a central event in regulation of glycogenolysis. In addition to this, we should know that epinephrine also acts on alpha-1 receptor in the liver tissue. And the activation of alpha-1 receptor causes stimulation of phospholipase C. Activation of phospholipase C results in increase in intracellular calcium level, and the increase in intracellular calcium level directly activates glycogen phosphorylase kinase. Recall that the mechanism is that phospholipase C catalyzes the hydrolysis of phosphatidyl inositol biphosphate with formation of two end products, inositol 3-phosphate and diacylglycerol. Inositol 3-phosphate then diffuses through the cytoplasm and interacts with inositol triphosphate receptors on the endoplasmic reticulum, and this provokes endoplasmic reticulum to release calcium into the cytosol. So this results in increase in intracellular calcium level, and increase in intracellular calcium level activates glycogen phosphorylase kinase. And now we see that both pathways lead to activation of glycogen phosphorylase kinase. That's why it's the central and the most crucial event in glycogen phosphorylase regulation. And once glycogen phosphorylase kinase becomes activated, it phosphorylates and thereby activates glycogen phosphorylase. And in activated state, glycogen phosphorylase begin to degrade glycogen into glucose 1-phosphate molecules. Then glucose 1-phosphate converted into glucose 6-phosphate, and liver by glucose 6-phosphatase cleaves the phosphate group that results in production of a free glucose molecule. And free glucose molecules enter into the circulation, and by this income of glucose molecules into the blood, liver corrects hypoglycemic state and maintains normal blood glucose level. In terms of skeletal muscles, we should realize that muscle tissue cannot maintain blood glucose level. So logically, skeletal muscles are not affected by glucagon. Muscle tissue requires glucose just for own needs, and the time of need comes during physical exercises. Because with physical exercises, muscle tissue requires a lot of glucose molecules for glycolysis, and glycolysis will provide energy for muscle contractions. So during physical exercises, glucose level in the muscle tissue progressively decrease. So muscle tissue has to use the additional source of glucose molecules, which is glycogen. So as we said, muscle tissue do not have glucagon regulation. Also important that skeletal muscles do not have alpha-1 receptors, but skeletal muscles have a lot of beta receptors that become activated by epinephrine. So skeletal muscles have epinephrine-mediated regulation, which is logical because physical exercises provoke increase in epinephrine level. Epinephrine stimulate beta 1 to 3 receptors. Stimulation of beta receptors cause activation of adenylate cyclase that activate protein kinase A. Protein kinase A activate glycogen phosphorylase kinase that phosphorylate and thereby activate glycogen phosphorylase. And glycogen phosphorylase will release glucose 1-phosphate molecules from glycogen. Then glucose 1-phosphate is converted into glucose 6-phosphate, and glucose 6-phosphate molecules will be the fuel for skeletal muscles during intense physical exercises. So basically from 3 pathways in the liver tissue, only epinephrine-mediated activation of beta receptors is present in skeletal muscle tissue. But skeletal muscles have the alternative pathway. As an alternative to glucagon pathway, skeletal muscles have calcium-mediated pathway. Because in muscle tissue, glycogen phosphorylase kinase is activated by high intracellular calcium level. The concept is that with physical exercises to provide contraction, sarcoplasmic reticulum release massive amount of calcium into the cytosol. Because calcium is the essential element for formation of calcium colmodulin links that will result in muscle contraction. And during contraction, muscle tissue wastes energy, because during contraction muscle tissue degrades ATP to ADP and then ADP to AMP. So muscle contractions result in decrease in intracellular ATP level and increase in intracellular AMP level. Overall, the more calcium is released, the stronger is the contraction, the more energy it requires. So basically, the higher the calcium level in the cytosol, the higher the force of muscle contractions. But also the higher the ATP consumption. So cell has to maintain intracellular ATP level. And the substrate for ATP production is glucose. Thereby very logically that high calcium concentration activates glycogen phosphorylase kinase that in turn activates glycogen phosphorylase. And glycogen phosphorylase provides fuel in form of glucose that is necessary for muscle contraction. So this calcium-regulated glycogenolysis provides synchronization of glycogen degradation with muscle contractions. What we mean under synchronization is that calcium by activation of glycogenolysis will provide proportionate amount of energy to provide the adequate force of muscle contractions. So energy production and muscle contractions are synchronized, so it's very interesting mechanism. Obviously, during muscle work, muscle tissue consumes a lot of ATP molecules that are initially degraded to ADP and then to AMP. So increasing AMP molecules is an indicator that muscle work is still going on and muscle tissue requires more energy. Thereby more glucose molecules have to be released. So to control the energy state, skeletal muscles also use AMP as allosteric stimulator of glycogen phosphorylase that will result in activation of glycogenolysis. And in both liver and muscle tissues, insulin decreases the activity of glycogen phosphorylase. It's easy to remember simply because insulin has the opposite effects to glucagon. Insulin secreted in response to increase in blood glucose level. So insulin level is high when the amount of glucose in the blood is high. And obviously with high blood glucose level, there is no need to release additional glucose molecules by glycogenolysis. So insulin stimulates phosphoprotein phosphatase. With activation, phosphoprotein phosphatase dephosphorylates glycogen phosphorylase, thereby inactivating it. And with inactivation of glycogen phosphorylase, glycogen cannot be degraded. So it's a mechanism how insulin decreases glycogenolysis in both liver and muscle tissues.